Am I audible?
Yes, sir.
Yes, sir.
Okay. So the topic which I will be talking about is today is on eukaryotic gene expression. Now, what is um, the important? What are the eukaryotic gene expressions? On one hand, you have studied transcription, translation in prokaryotes, and you have also studied about transcription, translation in eukaryotes. In your MSc, what you will be studying is regulation of gene expression in eukaryotes. In prokaryotes, you had studied about the regulation of lac operon. Optional, you can study also the trip operon. But now we will be talking about regulation and overview first on gene expression in eukaryotes. This is where a basic research takes place. A lot of research is taking place. So what is gene regulation? Gene regulation, it is the process of controlling which gene in a cell's DNA are expressed. DNA are expressed. End product of expression is a functional product. And this can be for example, a protein, for example. Now in eukaryotic cells, we have seen that, or the multicellular organisms, say in multicellular organisms, an important thing is that if you consider two different cells, that is two different cells, they may express different set of genes, even though they have the same set of genes. This is very interesting because even though they have the same set of genes, or the same set of genes, or what you can say DNA, two different cells, they express different sets of genes. There are of course same genes which are expressed, but essentially two different sets of genes are expressed. So depending upon which set of genes are expressed, the properties and nature of that eukaryotic cell is defined. Another important aspect, another important aspect is in eukaryotes, gene expression involves many steps, which will be this. We will discuss what are the different steps of gene expression. Steps involves many steps, and because it involves many steps, gene regulation also takes place at different steps. But we know that this is an important point. Major step of gene regulation is at the level of transcription. So we know that in our body there are hundreds and billions of cells. Can anybody say how many cells are there in your body approximately? তো বলতে পারো কতগুলো সেল রয়েছে? তো ঘুমিয়ে পড়েছো নাকি? কতগুলো সেল থাকতে পারে একটা শরীরে?
Sir, 32.7 billion number of cells.
Into the power billion, 10 to the power 9. 32 বলার দরকার নেই, কিন্তু you have something billions of cells in the body, in the range of billions.
Yes, sir.
So you have cells in the range of billions. And now Each cell is similar to each other as well as they are different from each other. They have all these billions of cells, they have the same set of DNA. There are different organs. Different. So, different organs have different genes expressed. Now, a question is under the question that this answer, this is basically a statement. The question is that billions of cells have same set of DNA or genes. Why do they look different? Why do they function different? Along with a why comes the question is how also. How do they look different and how do they function different? What is the point of difference? Why it is a difference? So this is where the definition of gene regulation comes very handy. So the answer is gene regulation. This makes cells different. So what are the functions? What does gene regulation do? It answers the question how a cell controls which genes out of many genes in the genome are turned on. That is, whether they are expressed. So this answers these questions. Because of this gene regulation, different types of genes, different types of cells have different sets of genes expressed. So your liver, the genes expressed in your liver are different from the genes expressed in your brain. So let us go to let us see how. Let us look at a different color use করা যায়. Say this is where I'm drawing a liver cell here. and I'm drawing a neuron, nerve cell. This is a liver cell, and this is a I'm drawing a nerve cell, for example. This is a nerve cell, a very bad diagram of a nerve cell. This is the nucleus. Now in a liver cell, you will find a protein and enzyme known as alcohol dehydrogenase. And in the nerve cell, you will find lot of neurotransmitters. You'll find lot of neurotransmitters. But in the nucleus, for example, let's see what happens in the nucleus. Here, this is the alcohol dehydrogenase gene. And this is, they say, for the example, the neurotransmitter gene. Draw it over here also. This is the ADH, alcohol dehydrogenase, and this is the neurotransmitter. So what happens here is that this ADH is transcribed and this is not transcribed. Neurotransmitter is not transcribed selectively. In liver cells, you don't have neurotransmitters. Here it is opposite. This is not transcribed, and this neurotransmitter, it is transcribed and finally translated, and you have the neurotransmitter present. So even though they have the same set of genes, both the types of cells have same DNA set, expression of ADH and neurotransmitter is different. In liver, you don't have any neurotransmitter being expressed, and in neuron, you have it being expressed. In the neuron, you don't have any ADH or alcohol dehydrogenase being expressed, but in liver, you have that enzyme being expressed. So this is what you mean by differential gene regulation. So gene regulation is different in liver, and gene expression regulation, it is different in liver and different in nerve cells. Similarly, you can say it is different for kidney cells, muscle cells, skin cells, it's different. Now there is the tricky question is, how do cells decide which genes to turn on or off? Now this is a very tricky question. We have seen that different cell types express different set of genes. However, two different cells of the same type, they may also have different gene expression patterns. So maybe two liver cells, they may have different gene expression patterns. Why? It depends on two factors: environment and internal state. Environment means the surrounding, this is outside, and this is inside. Examples from this is proteins or factors which are already inherited from the mother cell. Because of some reason in the mother cell, some of the factors, protein factors or regulatory factors or RNA factors or whatever factors which are present, it is different in the mother cells. So this will induce the mother cell will divide into two daughter cells. The daughter cells will inherit from the mother cell an altered expression state of internal factors. You can imagine, for example, in a zygote, how a zygote is formed. A sperm comes and it fertilizes the egg, but it is the maternal cytoplasm, the factors or proteins present in the maternal cytoplasm, which determine how the cells or what will behave in the future. So the maternal cytoplasm is the biggest determinant factor in making an organism or a human being or an animal. Although there is a contribution of the sperm, but that is a nuclear component. But how the nuclear factors are regulated, it is a determinant of the maternal cytoplasm. So this is an example from inside the cell, that is inside the maternal cytoplasm, and that depends on how it was expressed inside the mother's cytoplasm, how the proteins were expressed. Depending upon that, that's how the daughter cells will behave. This is example from which is coming inherent or from inside. From outside, you have chemical signals or growth factors. Growth factors. An important growth factor, is that fibroblast growth factor. fibroblast growth factors. So depending upon the surrounding, if it's in a region where there are liver cells, therefore the factors liver were determining growth factors which are present, it will decide whether this will express like a liver. Depending it is in the brain, it will express like a nerve cell. So let us see how cells, as an example, how cells respond to how cells respond to growth factors. So let us draw the cell. This is the outer membrane. Let's see if I can get a yellow color. This is, say, for example, the growth factor. Then this is the receptor. This is the receptor. This is the growth factor which we are talking about, external signal. And this is the receptor. What this will do is, it will activate different signal transduction, second messenger, third messenger, different components, signaling components. We are talking about signal transduction here. And eventually, for example, inside the nucleus, let's draw this. This is a, say, division promoting gene. And you have here the growth promoting gene. These are the promoters of the gene. They will encourage transcription factors to bind. So these are transcription factors. They will bind, and it will result in proteins. These are proteins which will promote growth and division. So this is how the exact things, few things which we can get from here. এখানে এটা রাখলাম, এখানে আমি এটাকে আঁকছি আস্তে আস্তে। So the cell detects the growth factor, এই যে growth factor in yellow, detects the growth factor through the physical binding to the receptor. ঠিক আছে, this is one point. Next point is binding of the growth factor it causes the receptor to change shape, and this triggers a series of chemical events. You can call it as signal transduction. And this results in activation of specific transcription factors. And what does the transcription factors do? TFs, transcription factors, they cause the transcription of specific genes. In this case, genes for growth and cell division. This is one aspect. So you can see it is the external growth factor which activates specific receptors, and this receptor causes a series of specific signal transduction events which will activate specific that growth factor, specific transcription factor. Those genes will be expressed because their transcription factors are activated, and these transcription factors, they activate they cause the transcription of particular specific growth factor specific genes. Now the next part which we go, that will be we can say that stages of, you can see that eukaryotic gene expression . can be regulated at many stages. Now we are talking about the different stages, multiple steps. There are multiple steps of a gene expression, so multiple steps of regulation. The first is chromatin accessibility. This means we know that the chromatin is organized in a higher order structure in chromosomes by structures known as nucleosomes. Correct? Beads on a string, nucleosomes. So now you can see that this chromatin, when it's bound like this, it is not accessible to transcription factors. So if a chromatin is methylated, we know that methylation, it condenses the chromatin, and acetylation, it relaxes the chromatin, makes it more accessible. So if the region of that DNA in that particular cell is methylated, it is not accessible to transcription factors. Not accessible to transcription factor means that gene is not expressed. So apart from methylation and acetylation, you have other factors like NRF, nucleosome remodeling factor, or the yeast Swiss-Snif complex, XWI-SNF complex. You have NRF, Swiss-Snif complex. These are all, they cause the nucleosome to move away, slide on the chromosomes, making this region more accessible to transcription factors. The next step of regulation is RNA processing. So what do you mean by this RNA processing? It is splicing, capping, and addition of poly A tail. So you have heard of concepts of alternate splicing. This is a very important thing you have read in your BSc, alternate splicing. You have different cells have got, although the same gene, for example, a particular gene has got, say, this is the exon 1, this is intron 1, exon 2, intron 2, this is exon 3. Say this is E1, this is I1, this is E2, I2, exon 3. So maybe in the liver cell, this is the whole, this part is spliced out, and you have only E1, and E3. This will result in another protein, a protein which has got the coding for E1 and E3. And say in the nerve cell, for example, you have both E1, E2, exon 1, E2, E3. This is a different protein. So the same DNA is giving rise via alternating splicing to two different types of proteins and resulting in two different types of gene expression in two different types of cells. This is one example. RNA processing, splicing and capping takes place. These are all the things. Then you have also RNA, messenger RNA, transport from nucleus to cytoplasm. In the cytoplasm, the mRNA can become a cytoplasmic RNA, where it is attacked by ribosomes, free cytosolic ribosomes, and there is translation, or it goes to the endoplasmic reticulum, where on the endoplasmic reticulum there is the translation by ribosomes takes place. This, there can be another level of regulation over there. Then the third important level of regulation is RNA stability. That is the lifetime of the mRNA molecule. Then you have this microRNAs. They chop off certain mRNAs. So if the mRNA has got a short lifetime in the cell before it is destroyed, then less amount of a particular protein will be formed. If it has got a long half-life, then a lot of amount of this protein, that same mRNA will keep on making many more proteins. If it's short, then it's destroyed, so less amount of protein will be made. So this is one aspect. The next aspect is regulation at the level of translation. Translation can be also regulated. Like transcription. Then protein activity. The pH or conditions in the cell is such that only a specific type of protein is expressed. For example, in your mouth, your salivary amylase, it works fine. But when it goes to your stomach, it's in the pH, it's destroyed. So proteins can be tagged with groups. One important group is ubiquitin. Once it is ubiquitinated, a protein, it is taken away for destruction. So by these happen differentially in different cells, and they cause a differential gene expression. So this is just the overview of gene expression which we studied today. So now we will go into details on each step. Next day I will start with the part which is on chromatin accessibility, and the transcription at the transcription factors and its regulation. This will be the two. Today we have understood what is gene, an overview of the regulation in eukaryote. So we have seen that regulation takes place at many steps in the eukaryote, and what are the steps? The first step is DNA accessibility, the chromatin accessibility. Second, level of transcription. Third, level of RNA processing. Fourth, level of RNA stability, the messenger RNA stability. Five, the protein stability. So there are 5 levels of stages of transcription of gene regulation or expression in eukaryotes. So on the basis of this, and then of course, finally, when everything is has been done, we will again go discuss a bit about in the prokaryotes and eukaryotes gene regulation differences. So one difference between prokaryote and eukaryote gene expression is that in prokaryotes, transcription and translation, they are closely coupled, that they take place together. When the mRNA is being made, immediately ribosomes bind and it starts to be translated there in itself. In eukaryotes, it takes place in different chambers. Transcription takes place in the nucleus, translation takes place in the cytoplasm, endoplasmic reticulum. So remember this difference between gene expression in eukaryotes and prokaryotes. This is perhaps the most important difference. It's compartmentalized. Transcription takes place in a different place. Translation takes place in a different place, in a different chamber. So this different, there is a level of increased step of regulation over here in eukaryotes, since it's a more complex organism. So do you people have got any questions? প্রশ্ন আছে?
স্যার
স্যার
প্রোক্যারিওটের কেসে ওটা কোথায় হয় বললেন, মানে ট্রান্সক্রিপশন আর ট্রান্সলেশন দুটো একসঙ্গে পরপর হয়, এটা কোথায় হয় বললেন? নিউক্লিয়াস?
আমি তো গ্রাম দেখাচ্ছি। In the case of prokaryotes, they don't have nucleus. They have got only one single cell. It's not compartmentalized. Prokaryotic cell, bacteria, there is no nucleus. There may be a nuclear region, something like this over here. So you have transcription taking place. Say, for example, this is the DNA. RNA polymerase sits over here, and this is what is being made. This is the DNA. This is the RNA which is being made. When the RNA is being made, on top of it, the ribosomes bind, and this is the protein is being made. So you can say that at one level, this is you are having transcription taking place. And here you have the translation taking place. So in prokaryotes, you can say that transcription and translation they are closely coupled. There is no other chamber like nucleus, cytoplasm. It's the same chamber. In eukaryotes, this is the nucleus. You have transcription taking place over here. Messenger RNA is there. Messenger RNA is exported to the cytoplasm. Processed messenger RNA. and here either in the cytoplasm, cytosol, or endoplasmic reticulum, you have translation taking place. So the mRNA is exported from the nucleus to the cytoplasm. In prokaryotes, there is no nucleus and no cytoplasm as such. So this is the this difference you should keep in mind. Any more questions?
স্যার বলছি এফজিএফ রিসেপ্টর এই গ্রোথ ফ্যাক্টরটা যে রিসেপ্টরে গিয়ে বাঁধছে ওখানে কি চেঞ্জ হচ্ছে, মানে স্ট্রাকচারটা কেন চেঞ্জ হচ্ছে স্যার, মানে রিসেপ্টরের স্ট্রাকচারটা কেন চেঞ্জ হচ্ছে?
ওটাই নিয়ম, কিছু বাইন্ড করলে রিসেপ্টরের স্ট্রাকচার তো চেঞ্জ হবেই। তোমার কানটা মুলে দিলে তোমার কানের স্ট্রাকচার চেঞ্জ হবে না? তুমি তোমার কানটা জোরসে মুলে দেবো, তুমি দেখবে তোর মুখটা খিঁচিয়ে দেবে, তুমি করে মুখটা করে নেবে, তাই তো হবে। কিছু বাইন্ড করলে তো তার স্ট্রাকচার চেঞ্জ হবে, সেই চেঞ্জ অফ স্ট্রাকচারটা ইট ইজ ট্রিগারিং দ্য ডাউনস্ট্রিম প্রসেসেস। বুঝতে পারলে?
হ্যাঁ স্যার।
তাহলে একবার কানটা মুলতে হবে তোমার, তাহলে আমরা বুঝে যাবো এক্স্যাক্টলি, আরো ভালোভাবে বুঝে যাবে। তুমি মুখটা তো বন্ধ হবে না, তুমি দাঁত খিচিয়ে মিশিয়ে নেবে তখন।
স্যার নিউক্লিয়াসের মধ্যে প্রোমোটারে গিয়ে বাইন্ড করছে তো?
ট্রান্সক্রিপশন ফ্যাক্টর বাইন্ড করছে।
আচ্ছা।
সিগন্যাল ট্রানজাকশন হয়ে ট্রান্সক্রিপশন ফ্যাক্টরটাকে অ্যাক্টিভেট করছে, ওই গ্রোথ ফ্যাক্টর রিসেপ্টরে বাইন্ড করছে, পরপর 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 স্টেপসেতে জিনিসগুলো একটা এ কে অ্যাক্টিভেট করছে, এ কে অ্যাক্টিভেট করছে। মানে কি হলো, তুমি হঠাৎ দেখলে তোমার একটা ক্লাসমেট কোনো একটা ছেলেকে রাস্তায় চুমু খাচ্ছে, এবারে তুমি কি করলে, এ বাবা কি কি করছে বলে তুমি আরেকটা বন্ধুকে শুনিয়ে দিলে, দেখ দেখ এরা এই করেছে। হ্যাঁ, সেও এ বাবা বলে আরেকজনকে শুনিয়ে দিল, এ বাবা করতে করতে আমার কাছে খবরটা চলে এলো, আমি গিয়ে ওই ছেলে মেয়ে দুটোকে ধার ধার দুটো থাপ্পড় মেরে বের করে দিলাম। এই হচ্ছে ব্যাপার। এখন তুমি যদি চুগলিটা না করতে তাহলে এটা হতো না। বুঝতে পারলে? দিস ইজ হোয়াট হ্যাপেন্স। এখন তুমি যদি সে ফর এক্সাম্পল তুমি যদি না দেখতে ওদেরকে, ওরা যদি তোমাকে না দেখাতো ওদের কীর্তিকলাপ, তাহলে তুমি এগুলোকে তুমি এই খবরটা দিতে না, তাহলে আমি ওদেরকে মানে বের করতাম না। কিন্তু যেহেতু তুমি দেখে ফেলেছো, তুমি হচ্ছে রিসেপ্টর, ওরা হচ্ছে গ্রোথ ফ্যাক্টর, পরপর গিয়ে আলটিমেটলি আমি প্রোটিন হয়ে ওই সেলের আমি ওদেরকে ফাইনাল অ্যাকশনটা নিয়ে নিলাম। এই হচ্ছে গল্প। বুঝতে পারলে? এটা হচ্ছে মানে খুব লেম্যান কথায় যাকে বলে বোঝালাম। এনি মোর কোয়েশ্চেন্স? আছে কোনো কোয়েশ্চেন?
না স্যার।
নেই? ঠিক আছে, তাহলে আমি নেক্সট ক্লাসটা উইদিন তোমাদের জানিয়ে দিচ্ছি, 2-4 দিনের মধ্যেই আমি আবার একটা বিশাল, এখন 15 তারিখ অব্দি জিনিসপত্র চলবে, অনুষ্ঠান চলবে, তারপরে আমি বলছি, তারপরে ক্লাস নেব তোমাদের। এই প্রত্যেকটা স্টেপকে পড়িয়ে দিলেই আমার কাজ শেষ। ঠিক আছে, ভালো থেকো সবাই।
ওকে স্যার, থ্যাংক ইউ স্যার।
ওয়েলকাম ওয়েলকাম।